All right, so let's talk about the lab that we did last week. So we were able to set up cells to do culture, and we set up a couple different cell lines, VHK, A549, HeLa cells, and MDCK cells. And we had some difficulties getting enough cells to do all the different lines, but we were able to get 6 dishes from the HeLa cell line and 6 dishes from the BHK cell line, that baby hamster kidney cell line. And so what we had was 2 dishes that were available to be counted for each time point, and we had 3 time points to count cells at. So what I did was I went in at 2 days, 3 days, and 4 days after they were plated onto the dishes and performed cell counts. What I did was I added trypsin to each of the dishes and then let it sit for 5 minutes, right, to resuspend the cells. And then I did a cell count just like you did with the hemocytometer. And so if you go to Canvas for this class and you go to Files, you can find that there is under labs images of the cells in the hemocytometers that you can use. So for example, here we have a folder called BHK, and then we have another folder called HeLa. So let's take a look at the BHK folder, and you can see that there's 2 dishes, dish A and dish B, for each of these time points, 2, 3, and 4 days in culture. When you look at dish A, for example, at 2 days in culture, you can see there are 5 images. Each image is one of those squares in the hemocytometer, right? So you can go in and count these cells, right? Just like you did in lab a week ago. So you'll need to do that, and you should enter all of that into an Excel file to keep track of your data. And then if you remember from the lab protocol, here we have a description of how to determine the concentration of cells, right? So that's going to be your concentration of cells. So you're going to go through that and do that for all those images. And let's say here's your Excel data. And let me put in one more spot here. Let's say that this is your each of the time points, 2, 3, and 4. And for each time point, we have a dish A and a dish B. And so you're going to get a concentration, right? And then that concentration, in order to get a total number of cells, you're going to have to multiply by the volume. So in that case, the volume that I resuspended those cells in was, let's see, half plus 1.5, so 2, right? So you're going to get a count or a concentration of cells, and you're going to multiply that times 2 to give you the number of cells that were in that dish, all right? So for example, I just made up some numbers here for us to use, right? So if you had 250,000 cells, for example, that you counted, right? If you multiplied that by 2, that will give you the total number of cells, right? So you're going to put down the calculations that you get for each dish when you're using this math, and that's going to give you the concentration of cells in the suspension. And then you're going to multiply that times the volume of the cells in the suspension, which I've told you here is 2 mils. And that's going to give you the number of cells in each dish, right? So here I'm just showing examples for the HBK cell line, but you'd have to do a whole nother set of analysis for the other cell line. So I've made up these numbers for us to use just as an example today. So these are the total number of cells that you've calculated, and now we need to determine the doubling time, right? So when we look at our notes from the cell culture lab, we can see down here that there is a calculation for determining the doubling time, right? So I'm going to break this down into little pieces for us so that we can calculate it easily in Excel without making mistakes. In each dish, you're going to take the number of cells and you're going to multiply that, the number of days those cells were in culture, and multiply that by 0.693, right? So for example, for here, I would say equals, this dish was in culture for 2 days, times 0.693, right? Next, you can do the same thing for the rest of your data, right, by clicking and dragging that little box. Now, if you refer back to that equation, we also have to divide it by the natural log of the number of cells divided by this original number that we had, right? So let's just break that down. The number of cells divided by the original number. So that's going to be the total number that you calculated for each dish divided by 300,000, or 3 times 10 to the 5th. Goodness sakes, yes, no commas allowed. Next, we have to take the natural log of that value. So you're going to say equal ln, left parentheses, highlight that number, right parentheses, and enter. And again, pull that down for everything. Now we can finally determine our doubling time. We're going to take this value over here, the days divided by 0.693, and divide that by our natural log of the number of cells divided by 300,000. So I'm going to say equals, and I'm going to select this number that we calculated, and I'm going to divide it by this other number that we calculated, and that is giving me a doubling time for each of these dishes. All right? So one thing that you can notice is that the doubling time seems to be on average about 3. It's not exactly the same. That's okay. If you were to plot your data, we talked again about what it might look like. All right. So if the cells are in this exponential log phase, if they're increasing at the same rate every day, you would tend to see the same number doubling time for every dish at every day. Right? So we're just going to assume that we were in this and that we can take the average, or that we were in this range. And so we are going to determine the average doubling time for each dish, for all of the dishes in the HBK cell line, right? So we're just going to say average and collect all these. Now my results, because I made up these numbers, they ended up being pretty similar. I planned it so that these numbers would represent that exponential phase. Maybe you get something different. Maybe they are widely different. That's okay. We're just going to average it and work with that number. Next, we need our standard deviation. So you're going to write equals STDEV, left parentheses. You're going to highlight all those doubling times that you got, right parentheses, and enter. Now you have the average doubling time for the HBK cells and their standard deviation. You would need to do all of this again for your HeLa, for the HeLa cell lines. And then ultimately, what you'll be able to do is make a bar graph, right? And I'm just going to make up some numbers. So I'll copy this one, and I'm going to make up a number here. Let's just say 3, right? I'm going to insert and just make a bar graph. So yours may not look like this, right? I've just made up data, but again, we've got a bar graph. We've got each bar is one of the cell lines, and on the y-axis we have the mean doubling time, right? So you'll notice that there's some problems with this graph. For example, Excel doesn't put in a y-axis. It doesn't give a title for either the x or the y-axis, and a variety of things. And in the rubric, I've incorporated some rules of things that I absolutely want to see in the graph or don't want to see. So just check out that rubric. Now we need to add our error, right? So let's just copy this data. Right, I'm just going to put it here, for example, and I'm going to make up a number for the HeLa cell line. All right, so this is the standard deviation for the BHK cell line, doubling time, and this is for the HeLa cell line. We're going to click on our figure, go to Design, Add Chart Element, and Error Bars. Within Error Bars, you're going to select More Error Bars Options, and down here on the right, you can choose Custom. Next, we're going to specify the value, and you're going to highlight both of these numbers for the positive error value and also for the negative error value. All right, so now we have a graph with error bars. All right, is this statistically significant? Is this difference real? Again, I made up this data, but I would say no. Look how much, even though their means are different, look how much their error bars overlap. I'm going to require that you perform a t-test in order to analyze this. There's a variety of different ways you can do t-tests. There's online programs. You can also do it in Excel. For right now, I'm going to let you guys take a stab at it on your own. Maybe you've done a t-test in other classes. Once I get to look at everybody's first drafts, then we will, I may put out a video if needed on how to do a t-test. All right. So if you did have a significant difference, you would want to put an asterisk, a little star, over the bar that was significantly different. Let's talk about the other things that should be in your paper. So I've created a Word document that has some guidelines. First of all, we've got our deadlines. You've got a good 2 weeks to do the first draft, and oh, I've got these mixed around, sorry. Final draft, due April 2nd. First draft, due March 31st. Now for this first draft, I will be using this rubric down here, right? And you'll notice that for some of these things, it says fully described and correctly described. I am not going to take off many points if you incorrectly described something because this is your first attempt. We're also all doing this online, all right? So for your first draft, I just need to see that you've tried to do each of these things, all right? But I won't take off points if you described it incorrectly. So say you described how to use the hemocytometer wrong, right? At least you tried to explain part of the cell growth assay protocol. If you did it wrong, I would just put in some comments on how to fix that. And then for the final draft, it should be corrected. So let's go back to this first page. The organization is pretty basic. You should have a title page. In the body of the paper should be the introduction, materials and methods, results, discussion, references, and then at the very end, your figure. Guidelines. So in the introduction, first of all, you need to have 2 references, right? At least one of them needs to be supporting your hypothesis. So you need to state your hypothesis, which cell line you think is going to have a faster doubling time, a greater doubling time, and why. And you must have a reference that supports that. And this is described in the rubric. The lab manual is not a source, so you cannot cite the lab manual. In the materials and methods section, you're going to be summarizing the protocols, right? Do not copy word for word from the lab manual, right? You should be able to summarize this. If you're having trouble doing that, what I would recommend is read the protocol and then write in bullet points the important things, and then take those bullet points and write them into sentences. All right, so your protocol does need to be written in paragraph form. It cannot be bullet points. The things that should be in your materials and methods are the cell type, the culture methods, so that would be like the media that you use, things like that. How you imaged and quantified the cells. So, what time points did you use for your growth assay? How did you collect the cells? What were they imaged with? How did you count the cells? I will post a detailed description of the microscope, its brand, and things like that that you can incorporate into the paper. And you should also talk about what analysis you used on the data, so a t-test. Right? Um, and the methods should be very brief. Most people make them too long, so don't kill yourself with the methods. Um, a lot of times I had to tell people to take stuff out, take stuff out. So before you even start writing your methods, look at this example paper that I've posted online. They have an example of how to write the methods for cell culture and also for measuring doubling time. And see how brief they are and try to replicate that. If there is any information that you did not get written down that you need, like what percent was the FBS in the cell culture, feel free to message your classmates, email them, and if worse comes to worse, you can always ask me, right? But I encourage you to email each other, whoever was in your group. The results section is going to state the data. What was the doubling time? Was there a statistically significant difference? And it must cite the figure. You should have, in addition to your figure, a paragraph of text summarizing your results. The figure alone is not enough for your results. You should also, you should not state the findings like, my hypothesis was correct. Whether or not your hypothesis was correct belongs in the discussion. Again, the figure itself is going to go at the very end of the paper. Next, we have the discussion. That is where you're going to summarize your findings. Was my hypothesis supported? Which cell had a faster growth rate? And then refer to one paper. So maybe your findings are in agreement with this paper or that, or they contrast with these people's work, right? Or maybe this study is a cool idea that we could do. We could do something like what these people did in the future. So you should have a paragraph or a section that talks about limitations of the study and future experiments. Your references should be in APA format. Your in-text citations should be used, also APA format. The references should be in alphabetical order, etc. The figure that we talked about, it's going to show the doubling time for both cell lines, and you can see the rubric for more details on formatting that. So again, I think this is pretty explanatory in terms of what you should have. This is exactly what I use to grade. All right, so use this as a checklist. If you have any questions about anything in this list, please feel free to email me. And again, I'm happy to make additional videos as well, explaining how to do different parts. All right. So again, best of luck, and I hope your classes are going well, and I'll talk to you later.